So now let's take a look at the systems that are involved in the immune defenses. The immune system itself doesn't exist in a defined compartment. Rather, it encompasses a vast network of tissues and fluids that permeate every organ and tissue. This allows for surveillance and recognition process and the functioning of a solid immune system. In the following section, we're going to be considering the anatomy of these main compartments and their roles in the second and third lines of defense. So the body is partitioned into several fluid-filled compartments. The ones that are of interest to us are the Reticuloendothelial System, or the RES, which is a network of fibers and phagocytic cells. We'll take a look at these in a moment, but they're basically a series of connective tissue fibers that allow communication between the various cells of the endothelium. There's the extracellular fluid compartment. That is the compartment that's outside of cells and contains the fluid in which cells are bathed. There's the bloodstream to consider, and then the lymphatic system. This yellow area represents lymphatic vessels. Lymphatic vessels are very closely tied to the arterial and venous circulatory vessels. Communication between each of these compartments is absolutely essential to proper immune system functioning. Regardless of which compartments originally exposed, everything is so tightly linked together that an immune reaction will be communicated to all others in a short amount of time. Let's begin by taking a closer look at the Reticuloendothelial System, or the RES system. It's a network of reticular fibers, as shown in purple in this image. These fibers provide a passageway between each of the tissues and organs. The reticuloendothelial system originates in the cellular basal lamina, and it interconnects cells, meshes with the massive connective tissue network surrounding all of the other lymphatic organs. Other organs consist of the thymus, the lymph nodes, the spleen, as well as lymphoid tissue that we see in the mucosa of the gut and respiratory system. Galt and malt are names for these. We'll cover them in more detail later. The reticuloendothelial system is loaded with white blood cells, especially macrophages. These macrophages are waiting to attack intruders in the skins, lungs, liver, lymph nodes, spleen, and the bone marrow. You can see these macrophages here. They just cruise around between the cells waiting to gobble things up. Let's have a quick concept check before we move on. What's the main component of our first line of defense? Is it phagocytes, antibodies, chemicals, or physical barriers? Well, we covered that a little ways back. Phagocytes we haven't quite covered yet, so we'll cross those off the list. Antibodies, we know, again, are part of the third line of defense. Chemicals were indeed part of our first line of defense. However, physical barriers are definitely our first line. However, we did see that there were some chemical barriers. So either way here, you could be correct. But most correct, I'd say, is physical barriers. Now let's take a look at the components and functions of the lymphatic system. The lymphatic system is a compartmentalized network of vessels, cells, and specialized accessory organs, which are designed to transport lymph and detect whether it's carrying any pathogens. As you may recall from anatomy and physiology, the lymphatic system returns about 15% of the extracellular fluid in the circulatory system proper. It acts as a drain-off system for the inflammatory response, so we get rid of excess fluids. It also renders surveillance, recognition, and protection against foreign materials. Now, lymph is the fluid of the lymphatic system. It's a plasma-like liquid that's formed when certain blood components move out of the blood vessels into the extracellular spaces, as it does in order to provide nutrients to the cells. And then it'll either diffuse back into the venous supply or it'll migrate into the lymphatic capillaries. Most of it goes back in the venous supply. However, the remaining 15% or so is left to go through the lymphatic system. The lymphatic system sort of acts like a detection system throughout the body. A small portion of each of that extracellular fluid is being returned to the heart through the lymph system. So it's being sampled, in essence, for anything that it might be carrying. that could be of danger to our system. So it transports a lot of lymphocytes, which are a type of white blood cell. It also is involved in transportation of fats, cellular debris, and infectious agents, which it will transport to the lymph nodes and other lymphatic organs for cleaning. The composition of lymphatic fluid parallels that of plasma, but it has no red blood cells. So let's look a little closer at these lymphatic vessels. They all follow the lines of our regular blood vessels, so our arteries and veins. As you can see here, they're closely intertwined with these vessels and ready to pick up any of that fluid. Now, the lymphatic system doesn't have a pump of its own, like the circulatory system proper does, where the heart is pumping blood out through the arteries, and there's still some residual pressure to push the blood back through the veins. The lymphatic system depends entirely on the contraction of skeletal muscles to return fluid into the circulatory system. You'll notice there's a high number of lymphatic vessels in the hands and the feet and around the areola of the breast. That's because these are great points of entry for microbes. The flow of lymph is in only one direction, from the extremities towards the heart. There is no outgoing lymphatic system. The lymph is returned to the bloodstream through the subclavian veins and into the heart. Ideally, it's been cleaned by this point. Now, the thymus originates in the embryo. It starts out as two lobes that eventually come together to form a triangular section of tissue that's right between the lungs above the heart. It has its highest rate of activity and growth until the age of puberty. And this makes sense because this is when an individual is gaining most of its immunological benefits. It shrinks gradually throughout adulthood, so we're less and less able to produce the T cells as we age. The thymus is really the site of T cell maturation, and T cells, as you'll see, are integral in the immune response. It's thymic hormones, or hormones that stimulate the thymus and help these thymocytes, which are essentially baby T cells, to develop their specificity and be released as mature T cells. After release, these mature T cells will migrate and settle into other lymphoid organs, like the lymph nodes or the spleen. So that brings us right into discussion of lymph nodes. The lymph nodes are small, encapsulated, bean-shaped organs. They're usually found in clusters along lymphatic channels and large blood vessels of the thoracic and abdominal cavities. You're probably most familiar with the lymph nodes that we see in the neck region, but we also find them in the axillary area or the armpits and inguinal nodes in the hips. The ones we see in our neck are the cervical nodes. There are many, many more aggregations of lymphatic tissue forming lymph nodes. Here's an image of a lymph node in figure A here. We can see basically that as lymph comes in through the lymphatic vessels, it comes in through these afferent lymphatic vessels. It runs through this general filter, and then it moves out through the efferent vessels. You may recall the details of how lymph nodes work from anatomy and physiology, but we won't go into them in too much detail here. Another organ of the lymphatic system is the spleen. The spleen is pretty similar to the lymph node, even in its structure, except it filters blood instead of lymph. Its primary function is to remove worn-out red blood cells. However, it's also involved in the immune function by filtering pathogens from the blood. And then it has resident macrophages, big white blood cells, that are involved in phagocytosis of these dead pathogens. Now, you may know someone that's had to have a spleen removed for one reason or another. And adults, it happens, can live relatively normal life without a spleen. However, if a child has to have one removed, they're going to be severely immunocompromised. It goes to show the role and the growth of the immune system, how important a period of a time childhood is to development of a strong immune system. Now we've covered the thymus, the lymph nodes, and spleen as our major lymphatic organs, but there are also many sites just beneath the mucosa where we'll see discrete bundles of lymphocytes, particularly in the gastrointestinal and respiratory tract. The positioning of this diffuse system provides effective first strike potential against the constant influx of microbes and other foreign materials that come in the food and the air. So this miscellaneous lymphoid tissue is found in our tonsils. You're probably familiar, there's a ring of tissues containing lymphocytes around the pharynx. These tonsils detect any pathogens that we breathe in or swallow. In women who are lactating or are pregnant, we see temporary sites of antibody-producing lymphoid tissues in the breasts. This makes sense because, after all, it's breast milk that's passing on many of the maternal antibodies. We also briefly introduced GALT, or gut-associated lymphoid tissue. This is our first defense against any pathogens that might be in the food that we eat. The appendix contains some of this lymphoid tissue. And then in each of the intestinal villi, where most of the absorption happens in the intestine, there are lacteals. Also, aggregates of lymphocytes in the small intestine, called Peyer's patches, are there to detect any potential pathogens so that we can send the message to the rest of the immune system to take care of these things right away. Some less defined patches of lymphoid tissue are in the mucosal-associated lymphoid tissue, which is associated with any mucosa, skin-associated lymphoid tissue, and bronchial-associated lymphoid tissue in the respiratory system. Again, these are a little less organized than the appendix and lacteals and Peyer's patches, but they definitely have a significant role in the immune notification system. So now let's take a closer look at blood. We'll be looking at how it's formed, what it's composed of, and the various functions of the different cells. The circulatory system proper consists of the heart, arteries, veins, and capillaries, as well as the lymphatic system. When we consider whole blood, we're looking at blood cells, or the formed elements, that are suspended in a plasma. The plasma is the liquid portion. So here, when we're looking at a sample of unclotted whole blood, if it's centrifuged, then a pellet will form in the bottom, an opaque pellet that consists of the red blood cells, and the plasma, the fluid portion, which is a yellowish, clearish substance, will float on top. Now, if we were to let the blood clot first and then spin it, we'll see that the pellet forms to the bottom, and then we'll have just serum on top. Serum is plasma without the clotting factors. You'll notice it's a little more clear than plasma. So plasma is composed of hundreds of different chemicals. The main component is water. That's about 92%, leaving 8% for all the other things, like proteins, such as albumins and globulins, which also include antibodies. There's a number of immunochemicals in there that we'll examine shortly, fibrinogen and other clotting factors, as well as hormones, nutrients, like our macromolecules that have made it across the digestive lining into our circulation, dissolved gases, oxygen and carbon dioxide, as well as waste products like urea. Now we're going to venture into examining the various types of blood cells, red blood cells and white blood cells and all the different subtypes. The reason that we're going to do this is so that you can understand all of the players in the second and third lines of defense. The production of red blood cells is termed hematopoiesis. It begins very early on in development in the yolk sac of the embryo. And red blood cell production continues throughout development, but is taken over by the liver and the lymphatic organs. And finally, we see that red blood cells are produced entirely in the red bone marrow. The active merocytes for hematopoiesis gradually recede as the baby develops. By the age of 4, we see that hematopoiesis predominantly happens in the ribs and sternum and pelvic girdle, the flat bones of the skull, and in the spinal column. So production in the large bones of the body has really ceased by the age of 4. The primary precursor of new blood cells are pluripotent stem cells in the bone marrow. These pluripotent stem cells go through a sequence of developmental stages that end up in the production of red blood cells, white blood cells, and platelets. We call red blood cells erythrocytes, white blood cells leukocytes, and platelets are called thrombocytes. Blood cells have relatively short lifespans. Really, they may only circulate for as little as a few hours, but most of them live a number of days. So here you can see one of the pluripotent stem cells. And there's a number of different stages that this pluripotent stem cell will go through in its differentiation to become a specialized cell with a particular form and function. Each of the specialized cells that we'll be considering and characterizing we'll find in this section of the figure. You don't need to worry for this class about all the other steps in differentiation. The first cells we'll consider are all in this yellow box. They're the white blood cells, or the leukocytes. They're programmed to develop into several secondary lines, and they're responsible primarily for all the different stages in immune function, which is precisely why we're considering them here. You'll notice that red blood cells, erythrocytes, and platelets have a relatively simple line of development. So here's an overview of the leukocytes, or white blood cells. First of all, we have granulocytes. And granulocytes have large cytoplasmic granules versus agranulocytes that have very small granules. Now, white blood cells are stained with stains that contain a mixture of dyes. These dyes can differentiate cells by color, or we could look at the morphology of the cells. The leukocytes appear either with or without noticeable colored granules in the cytoplasm. And they're split into the two groups, granulocytes or agranulocytes, based on the presence of these granules. However, scientists have also noticed that at higher magnification, even the agranulocytes have very tiny granules. So some have come to look at the nucleus shaped. We see that granulocytes have a lobed nucleus, as you can see here, and agranulocytes have much more rounded nuclei. There are three types of granulocytes that we'll consider. There are the neutrophils, the basophils, and the eosinophils. In the agranulocytes, we'll be considering the T cells, B cells, and monocytes. So let's take a deeper look. Beginning with the agranulocytes, we'll first consider the neutrophils. Neutrophils have nuclei which are in sort of a horseshoe or lobed shape. Often they're polymorphic nuclei, meaning they have many different shapes. They're the most common leukocyte and comprise 55 to 90% of our circulating white blood cells. They live about 8 days, but mostly in the tissues. They spend only about 6 or 12 hours in circulation in order to make it to those tissues. They're present in very high number in both blood and tissues, which will give you some insight into their importance in immune function. They function in the production of toxic chemicals, in phagocytosis of bacteria. In fact, those granules, as with all the granules, contain digestive enzymes. So once bacteria have been phagocytosed, then the granules release their digestive enzymes and break apart the bacteria. Neutrophils are the first to arrive during an immune response, during the phases of inflammation. Now, eosinophils. Eosinophils are present in the bone marrow and spleen predominantly. They attach to and destroy eukaryotic pathogens, and they're of particular importance in the destruction of helminth worms and fungi. These are the earliest cells to arrive at the sites of both inflammation and allergies. They work by attacking other leukocytes and releasing chemical mediators. So when an individual has either a fungal or helminth infection, we'll see a large increase in the concentration of eosinophils in the blood. So now our third type of leukocyte, basophils. We've had neutrophils, eosinophils, and now basophils. They're present in really low numbers in the body, less than about 0.5% of circulating white blood cells. And their function is very similar to that of eosinophils. Some basophils become mast cells, that is, they're localized. They become non-motile and bound to connective tissue around the blood vessels, nerves, and epithelia. They're especially important in allergic responses, as they're the first-line defenders against local invasions of pathogens. They are the ones that will recruit other inflammatory cells, and they're personally responsible for releasing histamines that we see in allergic responses. Now we move on to the agranulocytes. The agranulocytes include the monocytes and the lymphocytes. And the lymphocytes are broken down into two categories, the T cells, the T lymphocytes, and the B lymphocytes. Now lymphocytes are the second most common leukocyte. They constitute about 20 to 35% of the circulating leukocytes. And although they are the cornerstone of the third line of defense, we'll consider them here just so that we can compare all of the players in the game of immune response. Both B lymphocytes and T lymphocytes are transported to the bloodstream and the lymph, and they move freely between the lymphoid organs and tissues. They act to neutralize and destroy foreign substances. Now, the T lymphocytes are involved in cell-mediated immunity, and they mature in the thymus, so I remember them as T for thymus. The B lymphocytes, or B cells, are involved in antibody-mediated immunity, and they mature in the merocytes. I remember them as B lymphocytes, B for bone, where the marrow is. Another name for antibody-mediated immunity is going to be humoral immunity. Now, we'll consider both cell-mediated and humoral immunity as we move into the next chapter. But again, we're just introducing them here so we can get an idea of all the players on the immunity team. The second type of lymphocytes are the monocytes. They're the largest of all white blood cells. They're the third most common in circulation, constituting about 3 to 7%. The monocytes are like the cellular housekeepers. They mop up the messes that are created by infection and inflammation. They're discharged by bone marrow into the bloodstream and live as phagocytes for a few days. Then they differentiate into macrophages. Macrophages are the only exception to the short-lived lifespan of blood cells. These guys are relatively long-lived. They could live for years. They're responsible for many specific and non-specific phagocytic and killing functions. So we'll see them in this chapter and the next chapter. Because again, they are the trash picker-uppers. They process foreign molecules by presenting them to lymphocytes and saying, hey, this is some foreign stuff, go for it. They secrete biologically active compounds that assist and mediate and attract and inhibit immune cells and reactions. So they're key players in both specific and non-specific responses. Now, another form of monocyte are the dendritic cells. The immature dendritic cells move up from the RES, or the Reticuloendothelial System, and the lymphatic tissues. Here they trap pathogens, and then they migrate back to the lymph nodes and the spleen, where they become mature. In the lymph nodes and the spleen, they're now able to process and present foreign proteins so that other cells can become educated as to what are foreign particles. Again, involved in the third line of defense, so the topic of next chapter. So there are two types of monocytes, macrophages and dendritic cells. Finally, let's look at the non-leukocyte blood cells. There are erythrocytes, which are our red blood cells. They develop from the pluripotent stem cells in the bone marrow also. You can see their path of development here. They lose their nucleus just prior to entering circulation. So red blood cells, as you know, have no nucleus. And you're also probably familiar with the idea that they transport oxygen and carbon dioxide to and from the tissues. Now, platelets are another story. They are also formed elements of the blood, but they're not true cells. They're just parts of cells that fragment off. They function primarily in homeostasis and in releasing chemicals that are involved in blood clotting and the inflammation process. So now we've looked at all of the different types of blood cells, the leukocytes and the erythrocytes and platelets. Before we move on, let's have a quick concept check. It goes back a little bit. What would be the effect of lysozyme treatment on staphylococcus cells? I'll give you a hint that the staphylococcus cells are not necessarily important to this equation. Would they starve? Would they lyse? Would they grow? Or would they be unaffected? Really, we're asking you, what does lysozyme mean? Now, lysozyme is an enzyme that's involved in breaking things apart. So I'm going to go instantly to where we have lyse. They would lyse the cell. The relevance here is that lysozymes are often found in the granules of granulocytes, and so lysozymes act to lyse and break apart any of the invading pathogens.